All right, so we're on part 5 of our 7-part series of structural immunology. Oh, that is my PCAM. I got to make sure I pull up the right thing here. All right. So the immune system is more than antibodies. There's a whole branch of the immune system we haven't talked about yet, and it's going to be very important, especially this year, because we have Nick Maurice, who is a T-cell researcher who used to be in this class and now has a PhD and now is starting his own research and moving up through his career. He's going to talk about his research that he's done so far on T cells. And let me tell you, I've seen this research before. It's really mind-blowing, and it is pretty complex. And so what we want to do is we want to introduce you to the idea of what T cells are so that Nick can then ask you questions about it, and at least you've seen a T cell before. So that's the idea. So the important thing about T cells is they work on a different area of the body. The antibodies work outside of cellular systems. The T cells are able to work inside of cellular systems. That's because they have a little bit of help from the cells themselves. Antibodies can bind to pretty much anything. T cells have to bind to something that has a particular kind of protein called the MHC. And so we're going to talk about all that. The MHC is a window into the cell. When the cell is infected, its MHC protein will change what peptide it binds. And that is what the T cell can see. And so this is what the basic idea is that we want to explain to you. So I want to dedicate the person who basically started me on the path to even being a scientist. My grad school advisor was Craig Beeson. He was my professor at University of Washington, and then he moved to the Medical University of South Carolina and kept his lab going there. And he had tons of great lab work that involved things like heart attacks as well as immune system structural immunology. And so I was on the structural immunology side, and so I started learning about T cells from this. But I actually started working with the protein that T cells bind, which is the MHC protein. And I'm going to talk a little bit about what my grad research was. So remember that we went through a lot of trouble in that paper to get a surface antigen that would reflect an internal change in the cell. Even though it's a big change for the whole cell, it's very hard to find a good biomarker on the surface for the internal change of a RAS mutation inside the cell. Antibodies have a tough time with this because they're extracellular, but there are other types of the immune system that actually are set up to look for changes inside cells. They detect molecules that are called non-self inside the cells. They can detect basically what is normal for a cell, and when something is abnormal, it's not supposed to be there, it's not a usual part of yourself, then the T cells get activated and they alert the immune system to a potential problem. So antibodies are the easier ones to talk about. You probably remember hearing about those all through COVID. They're faster to respond. They're proteins. Remember that you can take them from one animal to another. You can store them just like you store proteins. T cells are mediated by cells. They're cells, right? So they have to stay alive, and you have to keep them alive. You can't just freeze them and wake them up later. You know, life is not that easy to turn on and off. And so because of that, T cells are more complicated, but there's also some arguments that they are more powerful. At the very least, they can do some things antibodies cannot do. And there are some therapies that are built around that. So remember that we start with the idea an immune response can be good or bad for your health. You can have an infectious agent, and you want your immune system to bind to and respond to that. That's protective immunity. If you don't have that, you have a deficient response, which is recurrent infection. If you have at the bottom a tumor, you want your immune system to bind to that, normal response. If you have a deficient response, the tumor survives and you get cancer. These are the cases where we want the immune system to turn on. But realize there's many cases where we don't want it to turn on. If there's an innocuous substance, we don't want it to turn on. A normal immune response to an innocuous agent is an allergy. You start responding and proliferating your cells in response to something that's not causing you an actual problem. And you know that that can happen. You know people with allergies, you probably have some yourself. If you graft an organ in, you want the person not to reject it. But if the immune system says, hey, this is not me, we've got to attack it, then you actually want to turn down the immune system in a case where you've grafted an organ into a person. And of course, your own organs, of course, you want your immune system to be able to recognize, hey, I should not activate against my own organs. And yet organs are complex things. Sometimes it can happen accidentally. When you have a normal immune response against a self-organ, you have a form of autoimmunity. And so just remember that there is a balance between the immune system turning itself on. I always see these things about boost your immune system, but I always wonder, well, what if you boost your self-reactions of your immune system? You boost your autoimmunity. And how do you know? You actually, if you're just boosting it in general, you'll get some bad effects as well as some good. And so you realize that the immune system actually has to be not too active, not too inactive. It has to be in the middle. It's supposed to, if you have tissue damage, then the immune system can actually cause that tissue damage. We don't want that to happen. But if you have an abnormal growth, you want your immune system to attack that abnormal growth, that is cancer. If the abnormal growth is from outside you, it's an infectious agent. You want the immune system to go after that. But if you have an interaction with the outside world, like an allergy, that the immune system acts against, you can have inflammation and tissue damage from there when you don't want it. So you see, all of these are about the balance that the immune system must strike. And also, whenever you have a vaccine, there is the possibility of side effects. And those side effects are just your immune system turning on against something that it shouldn't turn on against. With the vaccines, we're trying to get the immune system to focus on the particular thing that tells it what the pathogen looks like, and not against anything else. There's always fuzzy borders. So there's always going to be a little bit of danger when it comes to immunity and to vaccines. But we can minimize that danger, and we can talk about the experiments that do that. Okay, so there's a balance between these, and how the immune system maintains the balance is they communicate with themselves using molecules. And these molecules are called interleukins and interferons. So IL and IFN are basically little tiny subproteins. They're kind of like insulin sized. And the way that the immune system turns on and off different parts of itself and brings parts of it to a place where it needs to react against something is it sends these molecules through the blood that are these different factors that cause the balance to go one way or another. And the cofactors can turn things on, or they can turn things off. They can turn things on too much in autoimmunity, or you can maybe turn them off if you want to have a graft not be rejected. There's a whole lot of complexity about, and scientists like Nick Maurice deal with that complexity of how do the immune system cells talk to each other. For us right now, we'll just say that it's a balance. Okay. So I want to ask you right now, especially if those of you are going into the medical field, can you point to your thymus? It is an organ. It's a bit of an unusual organ. It's not a universal organ in all organisms, but it's very important to your immune system. And do you know what happens if you have a thymectomy, where you have the thymus being removed? Basically what you have is you end up having a deficient immune system. But you also can get along okay, because it's just one part of your immune system that's deficient. This is where your thymus is, so it's right around in the center. If you think you're pointing to your heart, you're probably pretty close to your thymus as well. And the reason it was called that is because it looks like a thyme leaf. Before they knew what they did, they knew what it looked like. And in fact, it was in 1961 where this was figured out. Realize that this is later than most of the antibody discoveries that we talked about before. Is basically they didn't know what they did, and only in the 60s did we figure out that the thymus involved the immune system and involved a very complex part of the immune system, which makes cells that grow rather than antibodies, proteins that flow through your system. In fact, some scientists were saying that the thymus was like the appendix. It had no real purpose, and you could take it out and people would be okay. Most people are okay, but you do end up with some kind of immunodeficiency that can show up in certain ways. By the way, that's the nature of immunodeficiencies. They're complex, and you don't really know when. You can know how severe they are, but you don't really know specifically how they will interact with things. Okay, so this Jacques Miller in London in 1961 removed the thymus from mice about 16 hours after birth, and then he found out that they had dysfunctional cells in their immune system. So he called the cells that were related to the thymus, that were deficient in the thymectomy mice, he called those T cells, for T for thymus. And it turns out it's right. The thymus is where these T cells gather together. Now realize that these are actually related to B cells. And in fact, if you look at these lymphocytes, you can see that there's 3 types of T cells that they show on this. There's the B cell right in the middle, because that's always front and center. You know, that's like the star of the show. But really, you have more different interesting types of T cells. And then you have that guy with the headband off to the left. That's a natural killer cell. And those are the ones that I actually worked on. Unfortunately, not much time to tell you about those today, but we will talk about T cells, which I worked on a part of those as well. So we have these different types of T cells, and notice the different outfits they're wearing. You know, you have the regulatory T cell, which has a police outfit, and you have the cytotoxic T cell, which has like a ninja outfit on. And so they have different roles, and we'll talk a little bit about what those roles are. Again, this is a really high-level sort of view of it. So, and I want to check to see how I'm doing on time for this. Okay, we are doing all right. So the thing is, these different T cells, they're not really wearing the different costumes, so you can think of them that way. The different T cells wearing different costumes, they're active at different points after the infection. Some of them are first responders. Some of them are sort of holding back until the first responders are done, and then they come in and they do their work. And really, there's 3 levels of infection that you can think about. After you're infected, within the first week, you don't have time to make new antibodies. So your pre-existing antibodies are there, and you have innate immune cells, like that natural killer cell I was talking about, that are able to react right away. They're not very smart about it. They have some set ways, and they have things that they always react against. They aren't able to adapt. 7 to 10 days after the infection, you actually start to develop adaptive cells, which can be either T cells or B cells, that have specifically grown to recognize that particular infectious agent. This is really amazing and cool, and part of this is the clonal selection that we talked about before. It takes about a week to do effective clonal selection and to multiply the B cells and to get them there. You also know that you also have a kind of response, so that is usually the response that kicks out the virus. And in fact, it's shown in red on the far right of this graph. You have T cell mediated killing of infected cells. So the very first, you don't have the T cells until, about a week after the infection, because it takes the T cells time to learn and to expand the ones that have learned how to recognize the infection. There's also a third speed of T cells. After the infection is kicked out, then you have a memory of it. And there are specific memory cells that are both B cells and T cells that basically, if they were useful once, they will hang out and they'll be called out of retirement when that infectious agent shows up again. These are called memory cells. And this can be months or even years later, depending on who you are and how your immune system works. So just remember, we have these different types of leukocytes. We have the macrophages, which we talked about very early on. Then we talked about B cells. And now we talk about T cells. And basically, lymphocytes means lymph-moving cells, lymph-associated cells. Within the T cells, notice that we have 2 major categories. We have ones that are cytotoxic. You can take that apart. They're killing cells. Then you have ones that are helpers, which don't kill cells directly, but they recruit other parts of the immune system that are armed, that are able to be cytotoxic. And this is just the tip of the iceberg. But let me tell you that B cells and T cells, the good news is they do both come from the bone marrow. The T cells are just educated in the thymus. All of them are born in the bone marrow. And you see over here, you actually see the B cells and T cells way over. A bone marrow cell can become any of these types of cells. It can even become a red blood cell, which is all the way on the right side of this. The same kind of cell could, instead of becoming a red blood cell, if needed, it can shift over to the left, and it can become a lymphocyte, which can be a B cell or a T cell. A B cell has a receptor on its surface that looks like an antibody. When that receptor is engaged, the cell is activated, and it starts to make more of that antibody. You see the plasma cell in yellow in the very corner. A T cell, on the other hand, has a different shape of receptor. You can even see it on this graph, that there is a totally different shape to the T cell receptor, and it's not totally different, but I'll show you how it's similar and how it's different. Basically, the T cells are activated, and then you have the cellular immunity from them. Finally, over here, macrophages are like a distant cousin, but they definitely are related. All of these are related. So all the white blood cells and the red blood cells, they are all related to each other. And it's fascinating to think about how they develop, but we're just going to take them as there for right now, because we're going to talk about the two branches. We've talked about B cells. Now let's talk about T cells. Now, if you just look at the white blood cells, we take the red blood cells off of it. You can see the same kind of family tree. You can see the natural killer cells are a cousin over there to the B and T cells. And you can also see a memory cell down here underneath the B cells. Basically, just like any family tree, you can take this apart and go into more detail. You can classify different things. This is a way that we understand things, and the general outlines are definitely what we need right now. It's interesting about each of these types of T cells. The memory cells are a different kind of T cell than normal effector cells. And it turns out that one of the ways in which they're different is they even have different metabolism. And this connects to our Biochem 2 topics. Naive T cells will proliferate when there's an antigen there, and they'll become effector T cells. And they will move from catabolic metabolism to anabolic. Then after the infection's gone, a select few of those T cells will become memory T cells, and then their metabolism switches back. They switch back to catabolic metabolism. So that's one of the reasons why we talked about that in Biochem 2, so that now you can see this and you can say, oh, I can fit this in with my biochem knowledge. And if you're a molecular biologist, you can see there is one transcription factor that actually both turns the effector T cells metabolism on and is turned off when the memory T cells turn the anabolic metabolism off. Kind of cool. You can get into the details here. Here we're just saying that there's different metabolism for different cell types. In fact, we can go into a little more biochemical detail, and you can talk about immunometabolism. That's kind of cool. I didn't know that existed before I found it. But you can put your biochemical pathways from Biochem 2 together with the different types of cells that are running different parts of those pathways over time. For example, in a normal situation, we can represent all of Biochem 2 with this right here. There's a few things left out, right? But if you look at part A, you have glycolysis, glucose coming in, you have the Krebs cycle in the middle, and importantly, you have glutamine coming in from the bottom. Glutamine is a different carbon source with different effects. It comes into the bottom of the Krebs cycle rather than the top. And that means it's a different kind of food. You also have fatty acids coming in here. And so A shows you the general scheme of metabolism. B shows you how metabolism works and how it's connected, but you already know a lot of that. It just shows that in aerobic glycolysis, you're turning up the glucose. You're turning up the Krebs cycle as a result, because you're pouring in glucose and you're breaking it down, and you're getting all these other things as a result. That is Biochem 2. What brings it into immunometabolism is you start to say, I wonder how different types of immune cells look different with when different states of metabolism are there. And basically we have 6 different types of, actually 7, because we have the naive sort of default control up there in the part A on the upper left. The other ones are 6 more types of cells in different types of things, and they have macrophages, and they have memory T cells, and they have different types of T cells. And just notice that the different types tend to correspond to either red, which has pathways that are anaerobic and breaking things down, catabolic, or you have blue that are anabolic, and they don't tend to be running on a lot of glucose. They tend to be running on the aerobic turning of the Krebs cycle, and they are working on building things rather than breaking them down and just being as fast as it can to make energy and to grow and divide and multiply. So it's aerobic versus anaerobic metabolism. Memory T cells are sort of, if you're sitting back and you're waiting, you're going to be aerobic. If you're basically running, you're going to be anaerobic. That's actually a lot like your muscle cells. It applies the same way to these different types of immune cells. And so there's lots of fascinating differences we can get into, but I want that thing to be the idea. Even T cells have multiple kinds. We've just scratched the surface by talking about the couple of kinds. There's other kinds, there are different numbers for the T helper cells, and they have different metabolisms as well. If you look down at the bottom here, you have upregulated glycolysis in certain types of T cells, and then you have the TMs and the Tregs, which actually have fatty acid oxidation upregulated, but they don't have glycolysis upregulated. This is a different version of the same thing, and it's just saying if you really get into immunology and you know about the different types of cells, you can use your Biochem 2 knowledge to say what are the differences in the pathways that they're running. So all that is to say, the different T cells all do run on the same kind of T cell receptor, and those T cell receptors work in a distinct way that's important for you to know what it's like. So, and the good news is the T cell receptor actually looks like a third of an antibody. You see the antibody, remember, it has 12 domains, and it has 4 domains in each of the 3 parts of it, and the 2 hands of it are each 4 domain sections, right? And they have the variable regions at the very end. Well, the T cell receptor looks like somebody just took one of these arms and just took it and made it into a receptor. It's just like one little hand there. It has the same 4 domains as the antibody fragment, the FAB fragment is what they call it. It even has an antigen binding site, and that site even has 3 loops each. So you have the exact same, that's why I went into detail on antibodies, by the way, because we can take that same talk and we can say, hey, that's on a T cell receptor. It's no longer buy one, get one. You no longer have bivalent binding, and it's used for a very focused purpose. Not to bind anything at all, but to bind a particular molecule on other cells. And the way that works is that the T cell, you see the cytotoxic T cell right here, will bind that molecule on the surface of the cell. So if you have an infected cell, it's going to be making lots of viruses. It has a lot of viral proteins inside it. Now the viral proteins eventually bud off and make more viruses, but it's kind of hard for the immune system to catch them at that point. Wouldn't it be great to find the cell that is a virus factory and to shut it down, right? Well, the infected cells do have some signals. When they're infected, they've evolved some signals that they put on the surface that the T cells then come along and they detect these surface changes, and then they say, oh, you're a virus factory, sorry, time to shut you down. And they do their cytotoxic activities. So the question is, what is this cell surface molecule that surveys the inside of the cell, that brings things from the inside of the cell to the outside where they can be detected by T cells? And by the way, here's actual pictures of a T cell and a virus-infected cell. You see the T and the V on the gray side over here. They're kind of small. Hopefully you can see it. And you can see that there is a flat part between the T and the V, where the T cell and the virus-infected cell are coming together in an immunological synapse. There's a small gap between them, and that gap is bridged by T cell receptors on the T cell binding some kind of molecule on the virus-infected cell. And so if they can detect unusual proteins that are made by viruses, they can also detect unusual proteins that are made by cancer. And this is another way that T cells, this is why they study the T cells at the Fred Hutch, because you can even have cancer vaccines that could teach your T cells, hey, when you see this particular molecule, this is associated with cancer, so I want you to freak out. I want you to actually activate against it. And that's the idea. There is such a thing as a cancer vaccine, but it will be generally T cell mediated, because T cells are the ones that can look inside tumor cells. Technically, they can look at the protein that samples the inside of tumor cells, and then they get activated. So this is, notice that the way they see inside other cells is because each cell has sort of a conveyor belt that takes all the proteins inside it and puts little fragments of those proteins onto its surface. This is how it works. When a virus infects a cell, here's a little cell biology. You have the viral proteins in the cytosol being made, and you have a bunch of those virus proteins, and they self-assemble and they become little baby viruses, and that's bad. While that's happening, you have a bunch of viral proteins inside the cell. And so you have this yellow protein inside the cell as well. It's called MHC class 1. And it hangs out where all the viral proteins are being made. And there are little fragments. There's proteins that fragment all the proteins inside the cell, so that the little fragments, the little red dots, will bind the yellow MHC protein. and they'll form a complex between the peptide and the MHC. So this is not a full protein, it's a fragment, it's a peptide. But it binds the MHC, and then the MHC is on a conveyor belt. It gets pushed to the surface of the cell, and then right there, it's holding its little peptide up. And then the T cells can come along, and they can look at the peptides, and they can see, is this peptide a problem or not? That's how the MHC are windows into the cell. If the T cell has it somehow detects that this peptide is a problem, it will become activated, it will kill the target cell, and then you will not have that virus-producing cell or that tumor anymore, if everything's working right. So this is why I think of the cytotoxic T cells as police. They have a lot of regulations. They have an academy where they learn how to just use of force, hopefully. We are teaching that. We want them to be a well-regulated, and we want them to serve and protect, right? So the idea is that you have the T cells need to be tightly regulated because they have weapons. And they need to be ethical because they have weapons. And we need to teach them how to destroy the virus-infected cells, but to leave your own cells alone. That's what the thymus is for. This is their weapon that they have. This is cytotoxic T cell. This is the synapse, and you have the T cell on the top, the virus-infected cell on the bottom, and you have the, if you look way over here, can you see this blue and orange interaction? The blue is the T cell receptor, and the orange is the MHC molecule. I know that because I even know what the MHC molecule's shape is, and the T cell receptor's shape is like that. And you can see that they're coming together, and that sends a signal to the T cell of unload your dangerous payload, discharge your weapons. And what it has right here in the middle, it has purple stuff. The purple stuff is reactive oxygen species, so that's a connection to a world from dust. It's things called perforin, which perforates the other cell. Granzymes, which actually are proteases or other kinds of hydrolytic cells that will break down cell walls, other proteins, and things like that. Basically, you've got really dangerous materials here, and they're all thrown from the vesicle in the T cell. They're thrown at the target cell. And you can even see the perforin perforating. Look down here. There's a perforin making a perforation. And so it's interesting, you can even do some biochemistry and try to detect what do these little vesicles look like? What do these weapons look like? And it turns out that some T cells can actually not just have vesicles that come out from inside the cell, they can actually package this stuff up into particles that leaves the cell and flies around for a little while until it attacks the other cell. These are just amazing. But these are like extracellular vesicles, but they have the same kind of stuff inside them. It's inside, it's the same thing. And so they call these supramolecular attack particles, and they are extracellular killing entities that deliver cytotoxic cargo targeted by the specificity of what's in their shell. This is kind of like a virus that's fighting for the good guys. I don't know. But you can even see with a certain kind of microscopy, you can look at the cell membrane surface. This is a target surface. You can see the SMAP coming up to it, and the particle, it looks almost like a virus, but it's going in, it's produced by a T cell, and it basically leaves it discharges its cargo in purple right here. You can see that it's going up to the membrane, pushing its stuff through the membrane, and basically killing the target cell with its toxic payload. So that is how MHC, you might have noticed that's MHC class 1. There actually is a whole nother branch of MHC called MHC class 2, because MHC class 1 is in normal cells, and it's running all the time. It's there to detect if the normal cell goes bad. Well, this same kind of system works really well. Wouldn't it be great to have this kind of system in a kind of cell that deliberately moved around and sampled the environment, so we wouldn't just be waiting for the bad thing to come to us. This would be going to the bad thing, and it'd be like a macrophage plus. And in fact, there are these systems on those type of immune cells. And so this is MHC class 2. The reason why I'm mentioning it is because it's basically the same biochemistry applied to a different purpose. It's just you have a macrophage in this case that's able to chew up the bacterium. What does it do with it after it chews it up? Well, it puts it onto a little molecule and puts it on its surface of its cell so it can activate certain types of T cells. That's really cool. This is a peptide binding a yellow protein just like before. So it's a peptide binding protein, but it is a slightly different structure, and it's a slightly different purpose in a slightly different cell. And so in fact, in B cells, you even have these MHC class II inside them, because if you think about it, how does a B cell know when it's gotten an antigen that is new? It just knows that it's bound something. How does it know that antigen is new? It asks for help from the T cells. The antigen antibody are drawn into the B cell. Then the antigen is chewed up, put on an MHC class II. MHC class II brings little snippets of that antigen out to the surface. And so the antibody waits for a T cell to come along and to say, oh, yeah, that's something new. We better make more of you. And it activates the B cell. So you see MHC class II plays a very different role. This does not induce cell killing. It induces immune activation, because these are good guys. These are immune cells that are trying to sample the environment, and they're wanting to check it against the T cells, which have a list of all the peptides that they've seen before. And if they say, hey, I've seen it before, they say, okay, you're okay, because I'm not going to activate you because I've seen that before. It's not that bad of a thing. We're used to it. If they see something they've never seen before, then they activate, and the whole immune system starts to go through inflammation, and hopefully the infection is fought back. So that's the basic thing. MHC class II molecules, you see it's on a macrophage here, and it's going to a T cell. The T helper cell is checking its list, and if it's not seen that peptide before, then it says, hey, you need to be more active. It activates it. Helper T cells activate their targets. They do not destroy them. That's why I think of it more as a radio. Maybe you can think of it as the police radio that is associated with the police that are the cytotoxic. Whatever it is, it's a communication. Not really. TH cells are not armed, but they activate, they speak in a way to the other cells. So, if you look at the T cell receptor, I mentioned this before, it's like 4 domains of an antibody, and it has, here's 2 chains right here. This is a T cell receptor. This is what it usually looks like. So biochemically speaking, the words we use for that are it's heterodimeric, it's membrane bound, you can see the little membrane tails at the bottom, and then it has variable regions and constant regions, just like antibodies did. The other thing you'll notice is it has little carbohydrates on it. Remember how in biochem we said everything on the outside of the cell has some sugar on it. That's a general rule. That's true for all of these extracellular proteins as well. And so if you look at the variable region and you look at the very end that combines with the antigen, you see the same arrangement as you see in antibodies. If you look on the left, you see the same 4 domains. If you look at it from the side, it looks from the side the same like an antibody. And most importantly, if you look at it on the right, you see that it's got 6 loops. In fact, it's got 8 loops because it's got 4th loops on the sides. But it's got 3 main loops, just like antibodies, even arranged in the same way. Even with loop 3 being the biggest one, just like they are in the antibodies. This is the same structure, adapted for different purposes. And this is the target. So this is an MHC molecule, and the one thing you know about them, they're supposed to bind fragments. They're supposed to bind peptides. And so it's going to look like that antibody that we showed before. It's going to look like a hot dog in a hot dog bun. MHC class 1s look like this. This is one of those cases where you have the hot dog bun that's really huge, and the hot dog is like, half the size of the bun, and you're like, well, who arranged this? But in this case, it's not about hot dogs, it's about the MHC protein doing its job, and so this works just fine. But just realize that that's what it's like. You have a smaller peptide in MHC1, and they're also called HLA proteins, by the way. You might run across that. In this class, I'll call them MHC, but you might run across HLA sometimes. So we said before it's a window into the cell. Here, structurally, it's got a groove, and the peptide fits into that groove. If you look at the side, you can see that there are all of these up and down, all of these, even this has immune domains underneath it. But the top domain looks nothing like an antibody domain. It's just a platform with two alpha helices, which we've not seen in antibodies pretty much. The two alpha helices are the two sides of the groove. The peptide fits within them. This is what I did my graduate research student work on, where we talked about how do these work as proteins, which parts of the structure are really important. So you have the peptide binding site, and for both MHC class 2 and class 1, they look pretty much the same. You have a helical peptide binding site on top, and you have underneath, you have more antibody domains. And when the T cell receptor interacts with the MHC, guess what? It interacts with both the MHC and the peptide. Some loops contact the MHC, some loops, especially the two in the middle, will contact the peptide. And so the loops on the MHC are just there to sort of snap the interaction into place. And then depending on the peptide structure, the TCR will decide, has it seen this before or not? If it's seen this before, it will not activate. But if it sees a new structure in the peptide, if it doesn't fit together right in a way that it remembers, then it will activate and the rest of the immune system will appear there. This is one of the earliest structures, but I like it because it shows the 6 loops in different colors. That you have the 6 loops of the TCR. It looks like an antibody coming down from the top. And on the bottom you can see the peptide. It's this little yellow worm between the two alpha helices. I love these structures, so I like talking about them. I'll try not to talk too much, but that's the basic idea. Now I have a question for you. Why didn't Wells do his thing on T cell receptors? Might be obvious to you. If it's not obvious to you, pause for a second and think about it. Try to come up with the answer. Why is it the antibot and not the T cell receptor bot? And so the reason is T cell receptors don't work by themselves. Antibodies work by themselves. So you can isolate and purify the antibody and you've got a functional protein. T cell receptors require the rest of the cell to be there. However, there are companies, you can't do it with an antibot type situation, but you can do it with a company that researches it to actually manipulate the T cell receptors on particular T cells. So T cell immunity is harder to work with, but it also in some senses is more powerful. It can look inside the cell, and the other thing about it is because it's looking at peptides, realize that it's only looking at the peptide residues that are going up. So it only has a few peptide residues that it's actually looking at. In other words, if the peptide residue that's going down is mutated, then that usually won't mess things up too much. Usually it'll present the same surface, and so the other protein can try to mutate, but there are certain mutations that don't affect the T cell's response. Now, of course, if you mutate one of the things it contacts, and it's an important contact, well, then that could get in the way of the T cell response, and evolution will favor those. But the thing is, that's a much smaller set because you have a lot of the peptide residues don't matter. And so if the organism mutates them, the T cells still work. That's what's cool about this. And that's why when we were talking about COVID, everyone was talking about antibodies, which of course they're the low-hanging fruit. They're easier to see. T cells were also important. And I think there were times when people sort of lost hope because the antibodies weren't working. But I also had the hope that the T cells were holding back, kind of like Gandalf coming in with the Riders of Rohan, whatever that is. I don't know if that works. But the idea is that the T cells are more complex, they take more time. But don't forget that they're there, because they are powerful. And there can be broad-based and long-lived immunity that are based on these little peptides rather than the intact proteins that antibodies bind. So in fact, how many different fragments of coronavirus does one infection of SARS-CoV-2 get your T cells to recognize? Turns out there's 15. There's at least, if you've been infected with SARS-CoV-2 at least once, then you've got probably at least 15 different fragments of coronavirus proteins that your T cells have learned to recognize, and then they will learn to activate against them when they see them again. This is one of the reasons why we've been able to build a wall. And vaccines, by the way, will boost this response too. Harder to measure does not mean non-existent. And so this is really fascinating things. There is some measure of cross-reactivity that happens with the T cells. Now we have to see what the bounds of this are, this has limits. It's not just like your immune cells suddenly become omnipotent and omniscient, and they're able to always see what's good and what's bad. In fact, if they were always turned on, then you would always have autoimmune diseases that resulted. So you got to realize that there is other things going on, but there are cases where you can have a peptide from something entirely different, and it will activate a T cell, providing immunity to you through that T cell to something you've never actually seen before, just because the peptides happen to be similar. And the peptides don't have to be very similar in their sequence, they just have to be a little similar enough in the surface that they present to the T cells. This is what the paper is that really started me thinking about this. This is from Mark Davis's lab, a big lab in molecular immunology. And what he did is he had a bunch of T cells that reacted to HIV peptides. And then starting from some anomalous observation in the lab, he started to notice these could be activated by things that immune system had never seen, that T cells seen HIV, but somehow it's reacting to this A. tropicalis peptide. It's reacting to this A. thaliana peptide, this P. marinus peptide. And you look up and down, you see these peptides, and they're all very different sequences, but to the T cells, they look the same. There's some kind of structural similarity between the HIV surface that's presented by the HIV peptide and whatever the surfaces are presented by these peptides. So the 5 clones are 5 HIV peptide recognizing TCRs. That's how they were brought up and recognized and grown up. But they found out that if you take weird bacteria that they never could have seen, you will always get a couple of these. For every single one of these, they were able to find at least one of the clones that reacted against it. That means that they are cross-reactive, and that a T cell against HIV could potentially protect you against A. tropicalis. Now realize that we're talking about a lot of different factors here, so it's not a complete protection. But we do have this idea of, as you get more and more T cells through life, you have more and more cross-reactive possibilities as well. And I do wonder, my kids seem to be sick a lot more than I am. Is it because my T cells that I've got give me a broader base of cross-reactivity that will fend off things that I don't even know that they're fending off? And then, of course, I'm going to get old very soon, and then my T cells will start to get weak, and then I'll start to be immunodeficient again. So it doesn't last forever, folks. This is another thing. So this is another way of testing it that Mark Davis did. So they noticed there were some similarities to the flu peptides, so they decided to have a hypothesis. They would say that, you have this flu peptide on the top 2, you have flu peptides, and of course you have donor TCRs that will respond to both of those flu peptides. That's what they're made to do. But they also noticed that the sequence matched this F. magna and the T. vaginalis that they have here. You can see that there's sequence similarity. And they were like, just based on our eyes saying these look like similar sequences, could these cross-react with the flu reactive T cells? And it turns out that they can. Only one case where they can't. And so by eye, you can look at a sequence and say, hey, that looks pretty similar to me. Even if they're from totally different diseases, it's possible that a T cell could look at them and also say, hey, that looks pretty similar to me. And this is a case where if a flu vaccine provoked immunity T cells against this particular sequence, those T cells would also protect against the other sequences from these other diseases based on the fact that they have similar enough structures. There's only so many structures that you can have on a peptide. So that because the fragments are limited, there is a sense in which they give you a broad base of immunity. And so this is actually shown in different ways. Now, we don't know exactly what mechanism always is for these things, but we can see that people who've had certain vaccines tend to be more protected against COVID, especially in terms of T cell immunity. It makes sense that the mechanism would be exactly what we're talking about here. So if you've heard that MMR or Tdap vaccine antigens will also give you T cells that will react against COVID, they found that in some cases that's true. Now again, this is probably less than half as good as actually getting a vaccine. So this won't really protect you, but it will help. And that is what you need to know about T cells.